Okay, so the first step is to get 0.25 grams of soil. So we'll add our tray, we will tare it, and we will carefully just add soil to this. So we get 2.25. Now that I'm done with this, this is our nitrogen-rich soil, and I'm going to add it to this little container with our bashing beads. Okay, so the next step is basically to add the bashing bead buffer that comes with the kit to the bashing beads and soil. And so you can see here that we have soil with our bashing beads inside there. Now I'm going to basically take our pipetter, we'll turn it to 750 microliters because that's what we're adding. Add a pipette tip. We'll grab some bashing bead solution here. Okay, and now we will just add it to our bashing beads. Alright, then we're all set and ready to go. Get rid of my pipette tip here. And there are a lot of expensive devices that go ahead and work with bashing beads. In this case though, we kind of have a makeshift one where we have our vortexer, right? We use this vortexer to mix cultures. We basically can take a piece of tape, put our bashing beads down on that piece of tape, wrap it so that it's around that edge on the bottom, and then I can just turn on this vortexer, speed it up to its maximum speed, and we'll run that then for 5 minutes. Okay, and what this does is it takes all those beads, bashes them into all the cells so that it breaks apart all of those cells so that it releases the DNA out into the bashing bead solution. Alright, so it's been 5 minutes. I'm going to take these bashing beads off of the vortexer. Okay. Next step then is to centrifuge because we want to get the all of this soil down to the bottom of this bashing beads assembly. So what I have is I have a second thing of bashing beads where I've put 750 microliters of water in this one. We're then going to load this on equal and opposite sides so that it's balanced weight-wise, and we'll turn this all the way up to 10,000 RPMs and run that for 1 minute. Okay. Alright, so now we're done centrifuging. So we'll open this up. We can look at our bashing beads and our solution here. We can see we have a supernatant on top that we're going to be carefully getting out of there. Okay. What I want to do is I want to put that into a 3F spin filter here. This is in a collection tube, and we're going to add the supernatant into here. So we want 400 microliters. So I'll go ahead and take my pipette and pipette tip out here. We have it set to 400 microliters. So now we will carefully take the lid off here, and I'm going to carefully then suck up .400 microliters from the top. I'm going to put the 400 microliters then into this spin filter. Okay. Now I need a weight on the other side. Usually in class you're going to have more students involved, so you'll have at least 2. But I'm going to add 400 microliters of water into another spin tube and another assembly so that when I centrifuge this, then it will have its weight on the opposite side. Okay, so now according to the directions, we need to then centrifuge this 3F spin filter in the collection tube at 8,000 Gs for 1 minute. So I couldn't do that on the other microcentrifuge, so I had to go to our big old reliable guy. This guy has a lid. I have to take off that lid there. And then looks like somebody left some old collection tubes inside there. So we'll take our sample, put it in a tube, and to make sure that we get exactly opposite our weight in there on that tube, we have to put on the lid. Okay, close this, and then we're going to turn this on at 8,000 for 1 minute. And of course we need to plug it in. Very important step to get power in this guy. So we'll go start, and we will run that for 1 minute. Alright, so 1 minute is up. We'll stop that puppy there. Let it then go down. As soon as it's all the way down, then we will open this guy up and let it continue to finish off there. And spin. And then once it's finished spinning here, we can then remove this lid and we'll look at the spin filter. So, take this lid off. Here is our water one that I used as the weight. But here is our actual sample, where we can see everything that we want down is in the bottom of the collection tube. So we're going to get rid of this 2F tube and keep this collection tube. Alright, so here is our tube all ready to go. We now want to add 800 microliters of genomic lysis buffer and 95% ethanol. So here's our 800 microliters. We'll start with that one. Alright, we'll just grab this. Our 800 microliters in there of our genomic lysis buffer. Okay, we'll then switch our lid. We have to adjust this then to 400 microliters. And now we will add our ethanol. So 95% ethanol. And we are all set. Alright, so now I'm going back to the Zimo kit. Here's our 1C spin filter. I'm putting that into a collection tube. I'm going to make another 1C spin filter with water so that we can balance again the centrifuge. If there were two groups working, the two groups would work in coordination here. Okay, here is our sample. I want to make sure that I mix this up really well, so I'm just going to pipette it back and forth to mix it up really well, so when we're ready to go. Okay. Now, according to the directions, then we want to add 800 microliters of this solution into this spin filter right here. Okay. So I'm going to get a pipette tip. I'll go ahead and put it into this one right here. We'll basically take 800 microliters and place it into this tube. Okay, then I'm going to take another tip over here, and over here I have some water. I'm going to take 800 microliters of water and place it into this spin filter tube so that my microcentrifuge is balanced. And then we'll put this on one side, this on the other side, and then we're supposed to spin it at 10,000 RPMs for 1 minute. Alright, so I'm slowing it down. We just spun this for 1 minute. So we'll let it slow down slowly. Now, in this particular instance, then we're actually going to find the tube. We're going to remove this tube. Make sure I record that as number 5. We'll put this spin filter in a brand new collection tube. And then I'm going to take 800 microliters more out of our original tube here and put it into the same spin filter that we just spun down. So we're basically repeating the steps that we just did. Alright, so I'll do that. Put that across from 5 so that we're set. Close that off and we shall go for 1 more minute. Alright, so we're done with that minute. I'll open this up, find our collection tube. This is our one with water, so we'll get rid of that one. Here is our collection tube then. Again, we don't want that from the collection tubes. Here's our 1C filter. We will put it in a brand new, fresh collection tube. And then we're going to add 200 microliters of the pre-wash, DNA pre-wash buffer. Okay, so here, pipette tip again. And I want 200 microliters of my pre-wash buffer. So I'll go ahead and get 200 microliters and place that into this tube. We'll make sure that we remember the place that we put this tube, so we'll put it in slot number 5, because over here we need 200 microliters also of this water to make sure that we get the right amount in this other collection tube. Okay, so we will then get my balance out, and I'll use 200 microliters for our balance one, so that now number 5 has the one that we want, and our balance is all the way over at 13, and we'll run this for another minute. Alright, so it's finishing up here. In this case, we want to keep our collection tube, and we want to add 500 microliters of our wash buffer. So I'll adjust my pipetter to 500 microliters. We'll open this up, make sure we get the right one, which is on number 5. Okay. Then we will get a pipette tip and add 500 microliters of our DNA wash buffer. Okay, now everything's in that spin tube, right? So we're going to keep the spin tube, we're just washing it out. So we'll then add 500 microliters, put this back in number 5, and make sure we have a weight that's also going to be 500 microliters. Okay, so I have 500 microliters of water here. That is going to be our balance over here in 13. So we'll make sure that we don't make a mistake there. And then we're going to spin this for 1 minute. Alright, now we're ready for the next step. So we'll go here, go to number 5, make sure we get the right one. Right, now we'll discard this collection tube. Alright, we'll put it up here with all our discards. Now the kit, or the instructions say use a microcentrifuge tube, so this isn't part of the kit, so I just have to use a normal standard microcentrifuge tube. Put that into our microcentrifuge tube, and then now I have to do a different amount because it's only 20 microliters. So I had to use this pipetter, which is a 20 to 200 microliters, set it on 20. We have different pipette tips here, so we will then get these pipette tips, and we need 20 microliters of the DNA elution buffer. Okay, so I'll get 20 microliters here of our DNA elution buffer, and I will put those 20 microliters now down inside our spin filter. Okay, and then I'll also need over here our collection tube. We'll put this spin filter in here as well, and then I'll put 20 microliters of water to make sure that it's balanced out appropriately. Alright. Now, interestingly, we'll put these two microcentrifuge tubes over here. The one we want, we'll put in number 5 again. And we'll put that lid there. We'll try to balance this, so we'll put this across in number 13. And then we only spin this one for 30 seconds. Okay, so 30 seconds at 10,000. So we'll open this up. So we'll go here, and this is number 5. We'll take this guy out and close this microcentrifuge tube cap right there. And we will then take this guy out and put him on the discard, and we'll discard that. And now we have our microcentrifuge tube, and you can see that we have our supernatant down in the bottom. Alright, now to the side, what I want to do is I want to take our 2 micro HRC filter, and we'll put that into another collection tube. We'll have a second one identical for balance. We're then going to prep it with the prep solution. So we want 600 microliters of the prep solution. So I'll go here, get 600 microliters of this prep solution. And then I'll put the 600 microliters of the prep solution into this guy. Then for the balance, we need an equivalent amount of water. So I'll get an equivalent amount of water, and we'll put it into this guy. Okay, we'll make sure that we put this guy in number 20, and this one in number 8, exactly across from 20, to make sure that it's balanced. We have to put a lid on this one because it's a little bit more, gets going really fast, and that has to hold that in there. All right, we'll close this, start it, and we'll run it at 8,000 ppm for 3 minutes. All right, 3 minutes are done, so we'll open this up, remove the lid on this guy. Okay. Number 20 is the one that we want, so here is our collection tube. We actually want that spin filter, so we'll keep that spin filter right there. Okay, here is what we have for our other sample that has our DNA from our soil. We have a new microcentrifuge tube, and then we'll also have to make sure that we balance it. So we're going to take our spin filter, prepared, right, with our preparations splitter, put it there. We'll take our extra spin filter and put it there. Okay. We're then going to add all of this into there. Alright. So I will take a pipette tip. We'll grab all of this guy. There's not much of it, so better be careful. And then add all of this to this micro HRC spin filter. And then I'll have to just estimate, and we'll take water, and we'll estimate about the same quantity, and we'll put it into this spin filter for our balance. And then we will do this all the way up at 1600 G for 3 minutes. Okay? And so this goes up to 1300 RPM. We can't go to 1600, but we'll just leave it at 1300. Alright, so we'll put this in here at number, what was it? Number 8, across from 20. We'll put this guy in here at number 20. Alright, we'll put the lid on. And we're going to go all the way up to 1600 for 3 minutes. And in this case, 1300, which is max for, we'll do a little bit longer. Alright. Alright, so it's done. So we'll go ahead and open this up here. And we'll remove the lid. Make sure we go to number 20, which is the microcentrifuge tube that we want. So then we'll get rid of that 2HRC tube, and then we shall label this one with our DNA group number, and I will then take our DNA and transfer it to the refrigerator so that we can go on to the next step in a few days. Okay, and that's it.